Hello everyone, my name is Joseph Renier Drews, and today I will be talking about the introduction to my group project. To begin with, the human digestive system is a complex network of organs and specialized tissues designed to convert food into essential nutrients designed for survival. This process involves the systematic and breakdown of large macromolecules such as proteins, carbohydrates, and lipids into their simplest chemical forms. This allows them to be absorbed back into the bloodstream for energy, growth, and repair. Now, by examining the interaction between the parietal, chief, goblet, and enterocyte cells, we can see how the body successfully manages the hostile environment of stomach acid and powerful enzymes while ensuring the safe absorption of nutrients and the protection of the body's own tissues. That's all for me. Thank you.
Hello, my name is Janice Demonsi and my matrix number is 110283. I will choose the 4 specialized cells of digestive system, which the goblet cells, enterocyte cells, apparatus cells, and chief cells. Now I want to explain structural specialization and its functions. The first one is goblet cells. Goblet shapes is narrow-based expanded apical regions. The expanded apical region allows storage of large amount of mucus. Large secretory vesicles filled with mucus. Secretory vesicles enable rapid release of mucus by exocytosis. Well-developed Golgi apparatus for modifying and packaging mucus. Mucus lubricate the gut and protect epithelial cells from mechanical damage and digestive enzyme. We got shot the picture here. And the second one is enterocyte cells. Microvilli on the apical surface forming a brush border. Microvilli greatly increase surface area maximizing absorption of digested nutrients. Very large surface area due to microvilli. Microvilli greatly increase surface area maximizing absorption of digested nutrients. Many mitochondria that can provide ATP for active transport. The third one is parietal cells. Extensive intracellular canaliculi, deep fault of the membranes. Canaliculi increase surface area for secretions. Numerous mitochondria. Mitochondria supply ATP needed for active transport of hydrogen ions. Microvilli lining canaliculi. Increase surface area for acid secretions. The fourth one is chief cells. Extensive rough endoplasmic reticulum, which is RER. RER allows high level of protein synthesis, well-developed Golgi apparatus. Golgi apparatus modify and packaging enzyme. Numerous secretory vesicles containing pepsinogens. Secretory vesicles store and release pepsinogens.
For my part, I will explain the interaction between parietal cell and chief cell in maintaining gastric homeostasis. Parietal cells secrete hydrochloric acid, creating a highly acidic environment in the stomach. This low pH helps denature protein and act as chemical signal. Chief cell respond by secreting pepsinogen, which is an inactive enzyme. This acidic environment produced by a parietal cell convert pepsinogen into pepsin, the active enzyme responsible for protein digestion. This cooperation ensures digestion occurs efficiently while preventing damage to stomach cell. By separating enzyme secretion and activation, parietal cell and chief cell work together to maintain a stable internal environment in stomach.
The next mechanism is mucosal defense and pH regulation. While parietal cells drop the pH to near 2, the goblet cells sense the chemical environment along with chemical signals to increase the mucus production to protect the stomach from damage caused by acid and digestive enzyme. When the stomach lining is irritated by high acidity, chemical signal such as histamine stimulate goblet cells to thicken the mucus layer, and then goblet cells and surface enterocytes secrete bicarbonate into mucus to create a protective neutralization zone to prevent gastric from damaging epithelial cell membrane. And the last mechanism is nutrient sensing and absorption. Enterocytes communicate with other digestive cells by releasing gastrointestinal hormones such as secretin and lactokinin when they detect the presence of fats or protein, and the feedback control if the enterocyte in the duodenum sense that the kind is too acidic. They signal back to the stomach to inhibit the parietal cells' activity to slow down acid production until the current batch of food is neutralized and absorbed. This is how specialized cells maintain homeostasis. First, for pH regulation, parietal cells provide the acid needed for sterilization and digestion. Next, goblet cells ensure that some acid doesn't destroy the tissue. Second, epithelial protection. Enterocytes are held together by tight junction, and goblet cells secrete mucus that protect this junction. And lastly, enzyme activation. Chief cells ensure digestion happen on demand and relying on the environment.
This is the visual illustration showing the social network of 4 key digestive cells: goblet cells, enterocytes, parietal cells, and chief cells, while highlighting their independence. Goblet cells are like the quiet guardians of the gut. They produce mucus independently, which does not need for constant messages from other cells. But they are not isolated. Their mucus act like a protective blanket for enterocytes, shielding them from harsh chemicals and friction. So, while goblet cells don't need others to do their job, others like enterocytes depend on them for safety. The second digestive cell is enterocytes. Enterocytes are the nutrient absorbers of the intestine. They absorb nutrients independently once nutrients are available. However, they rely on goblet cells' mucus to stay protected while doing their job. Therefore, goblet cells and enterocytes have a close connection where enterocytes depends on goblet cells for protection so they can absorb nutrients safely. Next, parietal cells are acid producers in the stomach. They respond to chemical signals like gastrin, histamine, and acetylcholine, but once triggered, they act independently. The acid isn't just for digestion, it activates enzymes from chief cells. So, while parietal cells don't need chief cells to function, chief cells can rely on their acid to become effective. The fourth digestive cell is chief cells. Chief cells produce pepsinogen, the inactive form of digestive enzyme. They do this independently, but here's the twist. Pepsinogen needs acid from parietal cells to activate into pepsin, so chief cells are semi-independent. And from here, we can conclude that parietal cells and chief cells have close connection since chief cells rely on parietal cell acid to activate their enzyme.